So we've had a custom software program written that overlays and works with the JANIS software. It's called the JANIS controller. We can change between telling the controller that we're going to do a plate setup, which is indicated by showing a plate here, or a setup from tubes. In this case, we're going to do a plate setup. So when I hit start, the first thing that happens is the sample plate flashes, and then you need to go and enter in a barcode. So you just enter in the barcodes at each appropriate position. So once all the barcodes are entered in, it goes into the database and checks that that source plate is meant to go with those barcodes, and it knows from that database how to distribute the samples to the appropriate plates. This is what an assay plate will look like when you receive it. It comes pre-barcoded with a C barcode. It also has a human-readable label telling you what the assay kit is. There are two seals on this plate. There's a top seal that is white and an underneath seal that's aluminum colored. The top seal is done to protect that bottom seal for two ways. One, from contamination, and from the fact that when you stack these plates, you can pierce the seals. In order to use this plate, you need to carefully peel it off without touching this top surface, and from this point on you need to only touch the sides of the plate when loading it and moving it around, because you don't want to contaminate the top surface by touching it, because your JANIS needles, or even if you load by hand, will puncture this and could bring whatever's on the surface into the well. So that's why we take precautions to keep this surface as clean as possible, so we don't introduce contamination into our wells. She's right now scanning the barcodes to register them in the software and show that they're on their appropriate positions. She then will hit the start button, and the JANIS will accept all of those barcodes, and if they're registered correctly, it'll proceed to the next step. The first thing the JANIS does is flush to make sure there's no air in the line, so that it'll accurately pipette and dispense the 5 microliters we need for each sample. And as we're doing this, we sort of keep an eye on it to make sure that as the syringes are coming down, that you don't see any air bubbles. If you see air bubbles, you need to reprime the system to get those out. And then pretty much from here, it is ready to go, and you can walk away and let the JANIS just do the work for you. So right now it's rinsing the tips just to make sure that there was no contamination from aspirating the sample. It's picking up new tips, and it goes and picks up 4 samples at a time and dispenses them into the appropriate plate and in the appropriate location. A sample consists of 8 wells on the PCR plate, so you'll actually go into your source sample 8 times and dispense it into 8 different wells. It's the combination of those 8 wells that gives your profile, whether it be for mitochondria or our STR assay. With this setup, on one JANIS, you can set up 9 PCR plates in about 2.5 hours. So the JANIS just finished adding all the template to this PCR plate, and now it'll be removed to have a heat seal placed on it. She then places it on the heat sealer and hits the button, at which it'll put on another seal to cover all the holes that were generated by the JANIS puncturing the bottom seal. This is done in preparation for thermocycling. She'll then place it on the shaker. We shake them for 30 seconds to ensure that the template has mixed with the PCR master mix, and then we spin them down. So it's just a brief spin in a centrifuge to ensure that all the liquid's at the bottom of the well, so that there's no air bubbles and we get the best PCR performance. So once you've added the template and heat sealed your samples, you need to thermocycle them. You do that by placing them in a thermocycler like this. Then you want to close the lid until it clicks on both of these, and then you move to the computer and start the cycling there. You select the appropriate program, right-click on Start Cycler, and then you select the thermocyclers you're using, which in this case are 6 and 7. You select settings for all thermocyclers. You change the volume to 40 because we do a total reaction of 40 microliters. and then hit OK. You'll see on 6 and 7 on the screen, they show up as a red line indicating how much time there is left on the thermocycler program. As it progresses, the bars change to green, indicating that it's completed. Once you've started the thermocyclers, they'll start flashing green, indicating that they're running a program. When they're actually done is when they'll flash orange, and that means they're ready to be removed from the thermocycler. After removing your crude PCR plate from the thermocycler, you then spin it down to ensure that all the liquid is at the bottom. The crude PCR plate goes into Hotel 1 on the instrument and into an open position. In addition to the crude PCR plate, you need to put on a magnetic bead plate and an elution plate. The magnetic bead plate goes into Hotel 2, and the elution plate goes into Hotel 3. After placing your plates on the hotels, you then want to check that all of your reagents are full. There are 3 different reagents that are involved in the cleanup. The first reagent is used for the desalting. The second reagent is sort of a polish and cleanup and gets rid of any polymers. And the third reagent is your elution buffer. We also have a system liquid, water that must be full, and 2 waste containers that should be empty. For the TOF itself, it has its own CR3 buffer to use to help spray and nebulize the solutions, and it has its own waste container. The next step would be to set up the computer. So after starting the software, this is how it'll look. You'll have your control screen, and in the background will be the actual acquisition software for the mass spectrometer. To start the instrument, after loading all of your plates and reagents, you just want to click the Start button. It'll ask you how many plates you want to run, and you can just input anywhere from 1 to 15. The next question it's going to ask you is, would you like to run a system QC test? And at the beginning of the week, or if the instrument hasn't been run for a while, you want to say yes to this. What this will do then is actually spray amounts of CR3 through the mass spectrometer, so you can see that signal is being acquired and how your peptides are performing. So right now it's going to rinse the injector with CR3. So you have a combination of a wash station where you can rinse your syringe and also aspirate CR3 to rinse the injector as it's doing now. Now it's going to inject a sample for spray. The fact that the lights change from green to blue on the screen indicate that it's changed the voltages for spray. As you see in the mass spectrum that is being collected, there are 2 major peaks. This low mass peak is one peptide standard, and then this high mass peak is the other peptide standard. These are the 2 peptides that are used to internally calibrate every well, and it's a good idea to check their signal intensity and their balance. They should be close to 1 to 1, and you should have good signal to noise on them. You can load up to 15 plates, and that'll be about a 24-hour run. And one of the nice features is you don't have to load all 15 at one time. You can put the 3 you have on that are already PCR'd, and as more thermocycling, you can just add those as those come off. Then the next morning, you're able to go and analyze your data and check on the quality of your data, look at your positive controls to ensure you want to run the instrument the next day, and as well as your data is ready for whatever analysis it needs.